Hey, today we're going to finish up Unit 8.5, Section 2. So we're going to start with PCR, which is polymerase chain reaction. Okay, so the word polymerase you should know is an enzyme that adds nucleotides to make either DNA or RNA. Chain reaction means we're just going to keep doing it over and over and over and over. So the whole purpose of PCR is to amplify pieces of DNA. So amplify just means copy. So literally this is the method that you can take a single copy of DNA and you can end up with millions of copies of DNA within hours. Okay, so this was developed in 1993 by Carrie Mullis. So it is kind of an old biotechnique, but we use it all the time. So in forensics, they use it to amplify DNA from crime scenes. So that would be blood or semen. So it's really helpful when you have a small piece of DNA or a small amount of DNA. You can actually amplify or copy it so you have lots to work with. So you don't want to mess up the only sample you have and potentially destroy your evidence in a crime. For medical uses, we can use it to target genes, to figure out if somebody has a certain gene or to find a mutation. So really it's copying DNA, but it's copying only certain particular parts of the DNA. In my research, I used it to amplify regions of DNA that had antibiotic resistance. So I would test different parts or different samples from farms to see if that sample had an antibiotic resistant gene from the bacteria. So you can use this on any kind of DNA. Okay. The way PCR works is you are making copies of DNA. Okay, so you need the target DNA or the DNA that you want to be amplified or copied. Okay, you have to have primers. So just like in DNA replication, you want primers to stick to your pieces of DNA so that you can copy that certain region. So once again, you're not copying all of DNA, just a certain region. Okay. You're going to need a DNA polymerase so you can make, because we're making copies of DNA, so we need an enzyme to actually make the DNA. So we use an enzyme called TAQ polymerase. So during this process, we're going to heat the DNA so it breaks the hydrogen bonds between the bases. So we want a DNA polymerase that can withstand that high temperature. Our DNA polymerase would denature. So we're actually using DNA from a bacteria, or DNA polymerase from a bacteria called Thermus aquaticus. So that's why we call it TAC polymerase, because it's TAQ, Thermus aquaticus. So it's an archaea bacteria, so it's used as high heat conditions. So if you are making new DNA, you're going to need some nucleotides. You actually have to add in what we call DNTPs for your A, G, Cs, and Ts. So how does PCR work? You put it in a machine, and the machine runs cycles. Usually it's about 25 to 30 cycles. So you take your DNA. The first thing you're going to do is you're going to heat it up to about 95 degrees Celsius, and that's going to break the hydrogen bonds between the bases holding the DNA together. Then you're going to cool it down, and that's going to allow your primers to anneal, or what we call stick, to either side. So if this is important because now we have a free 3 on either side. Our DNA polymerase can come in and fill it in, and we get 2 exact copies. So after one cycle, you get 2 copies, and then 4, 8, 16, 32. So after 25 to 30 cycles, you can have 1,000,000 copies of your DNA, which is really good to work with. Then you can manipulate that, run it on a gel. You can cut up with restriction enzymes. It just allows you to have more to work with. So that is the purpose of PCR. Okay, the next thing we're going to talk about is cloning. So a clone in biological terms is just any organism that has identical DNA. Okay, so what you do for cloning, you have the DNA of the cell that you want to clone, and then you have a donor egg. Okay, you're going to take the nucleus out of both of those. So if I want to make a clone of my dog, I would take one of her cells, take out the nucleus of her cell, because she's the one I want to clone, and I would put it into an egg of another dog that we removed its DNA. Okay, so my dog's somatic cells are already diploid, so when I put it into the egg, okay, which has had the nucleus removed, okay, the egg thinks it's fertilized, so it's going to start making a blastocyst and all that fun stuff, and it will actually implant into the uterus, and then you end up giving birth to an actual clone. Okay, so you're going to have to remove the nucleus from both an egg cell and your donor cell. The donor cell is the one that you want to clone. Okay, so you're going to take the nucleus from the donor cell, put it into the egg cell, which we removed its own DNA. So then the embryos are put into a mother. Okay, so the first mammal to ever be cloned was a sheep named Dolly in 1997. Okay, so we took the Dolly's moms, or the DNA we wanted to clone, out of her cells. We took an egg cell from another sheep, took its nucleus out, and then we put the clone's DNA into the egg cell. The egg cell then thought it was fertilized because it has 2 copies of the DNA already from her somatic cells. And then we grow up the embryo, and then we put the embryo into the uterus of a surrogate mother, and then it gave birth to an exact clone. So her name was Dolly. Okay, the third technique we're going to talk about are stem cells. Stem cells are cells in the embryo that can differentiate into any type of cell tissue or any type of organelle. Okay, so there are 3 different types, which we'll get into a second, but stem cells, you may or may not have heard that they're super controversial. When Bill Clinton was in administration, he actually banned the use of stem cells. If you weren't currently already using, you can't restart your projects. So there's a lot of litigation and legislation about that. It's been a big controversy because you have to get them from embryos. And the only way you can typically get them from embryos is if you make the embryo in lab or you get them from abortions. So that's why they're kind of controversial. So these are just some questions that stem cell research raises. So this is important for you guys to learn because this is always happening in voting. So you may have to vote on these topics sometime in your lifetime. So that's why we want you to know what they are. So these are just different questions that you need to kind of ask yourself when you're thinking about, do you agree or disagree with stem cell research, and why? There are 3 different types of stem cells. Okay, you have totipotent. Think of this as total. So these are cells that can make a whole total organism. Okay, so like totipotent is basically the whole entire total organism can be made from this type of stem cell because they're super early stem cells. Pluripotent, think plural. They can make many different types of cell tissue. They can be differentiated into a lung, a pancreas, a heart cells. So plural organs. And then multipotent means that you can have multiple cells, and they're usually only one type of tissue. Okay, so that's the most common because you can actually get them from your cord, from the umbilical cord. So some people keep these in case your child has cancer later on. They can actually use the cord blood stem cells as a treatment, kind of like a gene therapy. So those are your 3 types. And the last technique we're going to talk about is CRISPR. Okay, so CRISPR stands for clustered regularly interspaced short palindromic repeats. This was discovered back in like 2007 from a girl. So it's a way that DNA can be recognized by a bacteria as its immune system. So that was kind of confusing, but when viruses attack bacteria, they usually leave a little piece of itself inside of the bacteria's DNA, that one circular chromosome. So the virus infects its DNA, it incorporates it inside. When the virus leaves, there's a little trace left. So then bacteria have learned to use these gene editing enzymes to recognize that anytime that DNA comes back in, they attack that DNA. We are using those, okay, to actually cut DNA and modify genes in a living organism. That's a big deal. So the plasmids and all the other stuff that we've talked about before, you can do that before the organism's technically living because you can take the plasmid out, put it back into a living cell. This is actually a technique that you can actually edit your genes while the organism is completely living, which is revolutionary in this day and age. So big thing about CRISPR is you can edit genes while the organism is still living. There's tons of research on this. It's kind of a brand new technique. It's been around for a couple years, but for you guys, it's pretty new. If you want to know more about this, we can discuss it on Wednesday. You can do your own research, but that's all I have for section 8.5.